Good evening, everyone, once again. Hello, and welcome to this free webinar on OmicsLogic Bioinformatics Training. This webinar is jointly organized by Pine Biotech under the auspices of MedlabConvo in Nigeria. We're excited to introduce you to our OmicsLogic training programs. This program will help you see the role of big data and next generation sequencing technologies in life sciences. OmicsLogic Bioinformatics Training is designed for everyone interested to learn and apply data science skills, to study biological data sets in an effective and meaningful way. Before we get started, let me introduce you to Pine Biotech's vision and team. My name is Adetaya Boshade, and I am the OmicsLogic Community Manager for OmicsLogic Africa, responsible for daily interaction with hundreds of users that are part of our bioinformatics community, managed by Pine Biotech. Let me start by introducing Pine Biotech, the company behind OmicsLogic. I will share with you details about the upcoming OmicsLogic programs and explain how the training resources we will discuss today will be used in the upcoming data science training program. Dr. Alfred Tauber was the one that developed this in collaboration between Biotech and the Tauber Bioinformatics Research Center. What we will discuss today and throughout this program is a result of hard work by an amazing team led by the company co-founders, Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Alfred Tauber, who comes from a medical background, has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science and applications in medicine. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center at the University of Haifa and an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career, has developed multiple novel algorithm approaches to biomedical data analysis. The rest of the team also includes experts in bioinformatics, computer science, and training. And some of these are amazing team we're presenting today. Well, over the course of past several years, our team has been collaborating with a number of academic institutions around the world that have participated in content evaluation. Please, we kindly request that we should all mute our audio, please, to avoid interference. Thank you very much. Over the course of past several years, our team has been collaborating with a number of academic institutions around the world that have participated in content evaluation, curriculum review, and project design. As a result, the OmicsLogic programs have been thoroughly evaluated and are known to lead to excellent outcomes. Importantly, the programs are designed to support students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in bioinformatics, data science, and biomedical research. We are excited that this event has been led and organized by our dedicated team in Nigeria and India that brings together experts who are interested in data-driven research and have all recently completed our specialized training. What is the goal of OmicsLogic? The goal of OmicsLogic is to provide training in bioinformatics to enable independent research guided by mentors and peer examples. 2, to develop a growing community with shared passion for data-driven research and appreciation of citizen science. And our goal is also to enable students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. In this webinar, our team will introduce you to resources that will get you started on this journey. We do hope that you'll be able to benefit immensely from this webinar. Our objective solely is to get you started, to introduce you to our educational resources. And the very first one we'd like to talk about is the edutbio.info, which is a portal of educational resources. All you need to do is go to this website and get signed up. Let me take you on a few seconds journey to the website. I'm sure Mohit or Adela will type the website link into the chat options. But edu.t/bio.info. So here, you click on the register button. Here, you can put your username that you love to use on the website. You can put your email address. Then after you put your email address, you answer this "I'm not a robot" captcha, which when answered correctly, you can fill in your first name and your last name. Then after that, you just click sign up. Because I've already registered, let me just log in.
Yeah, so I've pasted the link. So if you can click on the link. So the screen is not shared, Tayo. So if you can share your screen.
Oh.
So yeah, we cannot see.
Sorry about that. Can you see now?
Yes.
Okay, let me take a step back then. All you need to do is type the website name. Once it opens, click on register. These are the options. You can type in your email address, a verified one. You can type in your username. You can decide on any username. Type in your email address. You have to fill this captcha. This "I'm not a robot" captcha. Include your first name, your last name, then click sign up. I'm already a member. So all I need to do is fill in my details, sort out this captcha, which at times might take 1 or 2 seconds. Then, so is there anyone that has successfully logged in? You can just press 1 into the chat box if you have successfully logged in.
So maybe if you can reload it, maybe in a different browser.
Yes, the captcha can as well, can take some time at times.
No, sometimes it's the cache, I guess. So
Yes.
because we're using it quite often.
So my login is successful now, and it's coming on now.
So did you guys try it doing the same so that we all come to the same place and check out the
Yes.
resources? So we'll wait for this for you to log in for a while. So please let us know that if you are able to log in or not. So please write 1 if you are able to log in, or if you have any problems, let us know. So this is the website, and then you can log in like Tayo showed us just now. Hello, Aisha. Were you able to log in? Aisha, you there?
Yes.
Okay. So, okay. Were you able to log in?
I'm still on it.
Okay, sure. Please take your time and let us know if you're able to log in or not.
Abdulatif, can you see the screen now? Okay, Frederick, thank you. Frederick said he's able to log in.
Perfect. So Tayo, could you show us the profile by that time?
Okay. So once you're able to log in, the first thing we would like you to do is to update your profile. This will allow us to communicate with you, recognize you, identify you as a member of our community. We also advise that you also add us to your social media links. So for profile, my profile is already filled. And from here, you can see once I log into my account, this is my full name. And beside it is my avatar, which is actually a picture. But for new registrants, once you log in, go straight to the profile. We can all see my screen. You click on the profile. And from here, click on the profile again. This is how a completely filled profile looks like. To have your name, this is it. You can edit if you've not added anything. You can edit. And here you can add your picture here or your avatar. You can add your full name. Here, this logo has to add your social media links. That's my LinkedIn account, which has been added here. This is for social links. Once you begin to do courses and finish and earn certificates, the certificate will also show here. But basically, here is where you can add your social links, your Facebook account, your Twitter account, your Instagram account. You can add them at the region of social links. Once your profile is completely filled, a completely folder has a picture or your avatar, has your full name, has your social media links, which will be imprinted here. Then here, any organization you join will be imprinted here. And as you continue in our program, the scores you get as you do your certificates and attend our webinars, both the free ones and the local ones will be accumulated and show here. So once you do this, the next thing to do is to continue to the courses page, where you can start taking advantage of our asynchronous courses, some of them which are mostly free, while others are paid. So, but to fully integrate us into the event today is one of our mentors, Dr. Moit Mazunda, who has a PhD in computational biology and has over 10 years of experience in industry and academia. I would like to hand over to you now, Dr. Moit. Thank you very much.
Thank you, Tayo, for the introduction and also for letting us know about the registration process for the free login and all those things. So let me just share my screen. So, am is my screen visible?
It's visible.
Okay, thank you. And Tayo, if you could help people mute themselves during the presentation, that would be great. Okay. Hello, everyone. My name is Mohit Mazumdar for everyone who's joining for the first time. And in my current role, I'm working with students and researchers and helping them learn and apply data science for biological problems. So I'm really excited to be a part of this training program as a project mentor. So today we wanted to discuss about bioinformatics. And so what is bioinformatics? Bioinformatics is a convergence of biology and data where both are equally important, as well as we see why this is in the next few slides. So who uses bioinformatics and who is it for? So the answer might have been statisticians, computer scientists, or engineers. But the truth is that more and more biologists and clinicians and students are now expected to understand and handle their own data to publish and succeed in both basic and translational research. So what we have realized is that biology itself is now rapidly acquiring the characteristics of data science, because data management and data generation is becoming much and much easier now. And the focus is on that what valuable information and insight that we can gain from the data. So this online program that we are going to do would be in which we would be talking a lot about big data challenges and opportunities, and we will be addressing those challenges and show you that what we are doing to address these upcoming technologies, implying these upcoming technologies to various challenges, various exciting challenges using high throughput data and high throughput technology. So what had happened and what has changed in the last few years is that the exponential growth of data. So if we go to NCBI, and I think many of you are aware of NCBI and some of the databases that are mentioned in this graph, and you can see that during this time, and many of the biologists and bioinformaticians uses the tool Blast is a tool to do a sequence alignment or a sequence alignment search for a large data sets or large databases that enabled us to do or understand many of this comparative analysis in bioinformatics. So starting from the time where Blast came up, from there the gene bank at NCBI, people started to doing more and more these experiments. And one of the reason was that the technology was getting cheaper as well at the same time. So with the technology available, and many of these projects were very important in the respect of human health, clinical research, and also like other aspects of agriculture and biotechnology. So there has been constant work from a large group of scientists, researchers, industry, who contributed in this data growth. So this big data, it can be described, it has some qualities. And when I am saying to you about big data, I don't know what you are imagining and how you're thinking about it, that how we can visualize big data. But it's different from what we have been used to. So we have been used to working on a single protein, maybe a group of proteins, group of genes, and we were all the time looking for an understanding that one important gene that we thought could be the cause of a disease. So during this time, we realized, after getting when this technology evolved and the Human Genome Project happened. So the Human Genome Project happened around somewhere from 1993 to almost 2003. So 1990 to 2003. So this during this time, I mean, we kind of sequenced and annotated the Human Genome Project, and then we realized that it's just not one protein or one gene which is responsible for a disease, but it's more complicated than that. So that's why big data started to play a role in this entire research, data driven research. And today we are going to talk more about that. So after this current talking about the growth that you saw throughout these years, and then the thing that had happened with these outbreaks, and we all know very much about COVID-19, and we all know about the SARS-CoV-2 pandemic. So this pandemic has actually shown this, that how things have changed over the time, and why we are now being able to gain such good insights, because we are not just able to record the data, but we are able to also analyze the data. So there's a lot of data that came out from this COVID-19. So today, what we are going to learn and what we are going to understand is about omics data. So how omics data is playing a role, what is NGS data, and how this is important for all of us to know from a perspective of a human being, from a perspective of a citizen scientist, from a perspective of a student, faculty, or a researcher. So the phenomics is the is the learning about the phenotypes. So we are looking at some of the features that we can visually see, and then how we are connecting that feature to a specific molecular event. So learning about the phenotypic characteristics, and then genomics. So in genomics, we are looking at the DNA. So we are looking at the sequence. We are look and trying to understand that what is the meaning, and why these certain mutations are so disastrous, and why these certain mutations sometimes helped us to evolve. So what is the meaning of these letters in the coding genome, and how can we understand and interpret the locations and the functions from this technology. Another important technology is epigenomics or epigenetics, which is about the non-genetic changes, which are methylation or different types of events, histone modification events that have been captured from chipset technology. And another very, very important omics technology is transcriptomics. So what we are learning in transcriptomics is gene expression. So we are learning about the gene expression, the expression of certain genes at different time points. That gives us a great insight about what is actually happening in the terms of what genes, what proteins are important for specific events. We can do comparison of different events. We can look for biomarkers. So all those things are part of this education and training that we are today discussing, and that we all wanted you to even to at least know about and be aware of. So when we talk about the omics technology, how it actually gives us insights. So we say a lot of things about using omics technology, using data driven technology, and then applying them into this research project. So how we start from a hypothesis to a research project. And when we are talking and discussing about this complex data, and everyone is inquisitive that what this data is, many of you are maybe beginners, so many of you have heard about these data types and what they mean and what can they be applied for. But in the end, I mean, all of this from a perspective of a biologist or from a perspective of a computer person. So in the end, what we are getting is data, right? So you see here, in this image, there's a table, right? And that table is showing the data that has been obtained from one of these experiments. So in the end, it's a table, or it's a data that is not structured, but we want to make it structured like this table so that we can do the analysis. So the challenge here, I mean, I was asking you to visualize the big data, and the challenge here is that when you are doing experiments such as the omics experiments, it's called the high throughput sequencing. And the reason it's being called high throughput sequencing, because you're getting tons of data. So you're getting data in megabytes, gigabytes, terabytes. So you obviously cannot open them in your Excel sheet. So you need a computational infrastructure to be able to analyze that sort of data and to be able to interpret and apply algorithms and tools, data science into these sort of data sets. It's really exciting, and it's not so difficult to learn, because we kind of believe in learning from the very beginning, from the basic. So that's why we started this collaborative program, and we wanted to start from the basic and then slowly move and grow towards the research part, and to learn in depth and to master some of these techniques and data analysis. So today we wanted to talk about our upcoming program with MedlabConvo, and as you saw that there's a big team out there that is there to support you all. So we are trying to support you all by providing you kind of right guidance. So the Omics Logic programs have different several components. So these different and components comprises of live sessions like this one. So we will have live sessions for the program that you all showed interest in, and those live sessions will be just a part of this entire training, because other than the live sessions there's a lot of stuff to look at, there's a lot of interesting things to do, and there's a lot of activity. If you can accommodate that sort of time, so you learn a lot from those experiences. So what we are offering along with the just the sessions is like a lot of materials that we have prepared for you, uh, graphical content of educational materials that includes videos, that includes text, that includes quizzes, assignments, that gives you an understanding and in-depth understanding of how to simplify the concepts and understand in a manner that you can apply it for research. So that's what we have been trying to do in our complex researches, and to bring it to everyone so that we can actually apply it in a regular basis and learn from it. So so uh, so we will just take a brief pause here. Sorry, can you hear me now? Hello, Tayo, you there? Am I audible?
Yes, yes, we can hear you loud and clear. You can continue,
Can I
please.
hear you now, Mohit?
Thank you, Sani.
Hello, Mohit.
Okay, I just got disconnected, I thought. So as I was telling everyone about the online courses, Tayo, and the online courses are associated with a profile. So you created a profile, and I think, Tayo, if you could please again write down the address, web address for the educational portal. So we just want all of you to go to the educational portal and create your login. So you will get a login, and this is for everyone. If you are joining the program, if you're not joining the program, you're going to benefit from it, because if you're not joining the program, then you will get an educational profile, and then you get all the free resources. And if you join the program, then you get all the paid resources, and then do the practical assignments as well. So the difference between free and the paid resources are basically the online sessions and the practical assignments and the hands-on activity. So the third part of this resources that we were talking about is the T-Bioinfo platform and the Codomics Logic platform. So we will be seeing those as well, and we will see how it is easy, it's easy to learn from these platforms and learn the logic of the analysis. So about the program, we kind of will be starting with an introduction. So about the introduction, I would like to like to know about all of you who have joined today's session. So we kind of thought to do some kind of polling, so in which we can ask the questions that we want to ask from you all, and then we will know more about all of you, and then we will discuss that how we can make it customized and make it more focused on your ideas. So I'm just launching a poll here for everyone. So you might see a poll in your chat, in your Zoom box. So please fill up this poll. So please let us know which country you are from. So if you're from Nigeria, if you're from Ghana, if you're from Zimbabwe, if you're from Egypt, if you're from Kenya, or if you're from some other country, so you can write it down in the chat box if you're from other country. And uh, yeah, so my country is not here as well, so I'd like to write others and India. Yes. So what is your educational qualification? That's the second question. So if you can see that. Yes. So bachelor's degree, are you doing, like, if you're undergraduate, or if you're pursuing a bachelor's degree, if you're doing if you're in a master's degree, or if you're doing a PhD, postdoc, or if you're faculty, or you're interested in research, or you're doing some research in some organization and you're a researcher, so you can write it down there as well. So once we have that, we will also discuss based on those areas. And the third question is, what is your level of bioinformatics knowledge? So have you done any bioinformatics courses before, or were there any courses in your college? Did you get any chance to do some sort of sequence-based analysis or some sort of project in bioinformatics? So are you a beginner? Are you familiar about it? Have you heard about bioinformatics? Do you know some concepts? Yeah. Okay. I will end the poll maybe in a minute or so. So please take your time and let us know. Okay, I see Jadi says his registration is successful. Thank you. Thank you, Jadi. And thank you, Editayo. All right, I am going to end the poll in 10 seconds. And thank you everyone for participating in the poll. And you can write down in the chat if you are not being able to, if the option is not there in the poll. So you can write it down in the chat as well. So Jackton, you're saying that I'm not audible. So Editayo, am I audible?
Yes, you're audible, sir.
Annie?
Probably you need to work on your system. Check the audio part of the system, most likely.
Yes, you're very audible.
Okay, thank you. Yeah, so okay. No, I have two logins. So one has audio, another does not. So that's why maybe it was showing like that. Okay, thank you. I'm just ending the poll, and thank you all for submitting the information. So as I was telling you that we will be covering in the first week when the program starts in a couple of days, we will be introducing you all to the computational tools and databases that are associated with the training, and that will take you to different data types. So an introduction involves what is the application of bioinformatics, or what is the application of omics technology, what is the application of data science in biology in these different domains. Like for agriculture, it's in the application in agriculture if you're interested in doing agricultural research, and if the infectious diseases, the application in infectious diseases, the application in oncology. So we are doing all those sort of research programs as well. So we will be discussing you in that session, in that during that entire week, that what is where you can apply bioinformatics to, what all research you can do, and where are these resources that you can learn from. And then in the end, we will be having a question and answer session. We have an interactive session where you can ask your questions, whatever your problems that you're facing. So one of the important aspect of this entire process, this entire time we will be doing this for 1 month, would be the support. So you can have all sort of questions. You can ask any basic questions that you want to have, and just let us know, and we'll be showing you that how we can be able to communicate with each other during this program through a page, through a website page, which will be a part of this program, which would be the organization page, which will be showing you in some time. But before that, I just wanted to tell you all that the part of this the program, the first part of this program will be about introduction and introducing you to the several resources that we have. So we have these three platforms, and then the sessions, and also the different data types. So the three platforms that we will be working on, all of us, when we start this program, are the educational platform, or the portal where you saw the courses. So there we will be finding a lot of other things than what we have shown you today. So Introduction to Bioinformatics is a free course. So once you are there on this site, and and I think we have been sharing this, and many of you have been able to log in. So please explore it when you have time, or please explore it now. So in the EduT Bioinfo platform, you will find Introduction to Bioinformatics course. So that would be the first course that we will ask you to take, so that there's also a form in the end of that course that let us know, let us understand, makes us understand that how you have gone through this course, and how comfortable you are with bioinformatics after learning from Introduction to Bioinformatics. So after doing that, we will be doing the hands-on exercises on server T Bioinfo. So let me take you to the second side, to the server T Bioinfo platform, and show you how it works. So this is the first platform, and you'll see that I have a user profile, as I was telling you. So there is one question. I believe I'm not late yet. So yeah, we are also recording this meeting, and we will be also emailing you the meeting, this recorded session of this entire video of this entire session. So I think it'll be fine. So if you have missed, you will catch up. So as I was telling you, that this is the first platform that Tayo showed us, and then the second platform that I wanted to show you all is the T-Bioinfo platform. So this is the platform that you will get access to after registration, because this requires this is a data analysis platform that has a powerful platform that has a lot of capability to do different type of big data analysis. So in our program, we will be learning about this platform. So it's a platform that is built for someone who has no knowledge, or who has a very low who has no background in bioinformatics. So we are kind of build this platform thinking in collaboration with Tauber Bioinformatics Research Center, who built this platform to help clinicians and researchers to be able to do their analysis with an ease, with a click of their mouse. So I'm going to show you how it works. So there are different sections here, and if you hover mouse, you will learn about that technology. You just have to hover mouse and read about it, that what it is about and what it says. So there are different sections. So NGS data type, we discussed omics technology today. So these are different NGS data types. Then we have different other data types like proteomics data, mass spectroscopy data, structural biology data, and then data integration and modeling. So not all of these tools we will be using for the program, because these are a lot of research tools that are used for different kind of problems. So we'll be exploring some sections of this platform, and one of the sections would be transcriptomics, genomics, metagenomics, that I'll be showing you in the next slides. And then we'll come back and show you how it works in the TBioInfo platform. So going back to the presentation, so the first session, as I was telling you all, the first session will be on 3rd February, and we'll be meeting at the same time. And the first session will include the basics of biology, and then we will be covering the basics of big data, and then the basics of next generation sequencing, and the basics of all this technology that we have been discussing in this session. So the first week will be to getting acquainted, for you to get comfortable with this platform, and get access to all the platforms, and communicate with us if you have any issues. The second week, we are going to dive into the first technology, which is genomics. So genomics involves understanding of DNA, right? We talked about that. So how these different technologies are helping us understand and develop such understanding which is helpful. So this entire spectrum, and before I say more about genomics, I would like to specifically tell you all that genomics has a huge application in clinical practice, and in patient care, and also in other applied fields, as I was telling you. So one of the major application of genomics is in precision medicine. So the what we are learning in genomics is the spectrum of DNA variations, which in human genome, which comprises of small base pair changes. You have heard of substitutions, insertions, deletions of DNA, then large genomic deletions. So we will be discussing all those in inversions and translocations. And then the understanding of genomics is pretty much important to learn about cancer, right? Because in cancer genomics, cancer is caused by somatically acquired mutations, and it's a disease of genome. So yeah, to learn cancer, I think it's very important to learn about the technology, genomics, and to understand that how those mutations are like causing. So in genomics, you will have access to 3 courses. Introduction to Genomics. We will start with Introduction to Genomics, which gives you a very basic understanding of what genomics is. And then Genomics 1 talks about these different algorithms. You can see here a pipeline. So we will talk about what a pipeline is. But before we talk about what a pipeline is, can anyone say and tell me here, do you know what is a pipeline, or what is these algorithms and this picture is? So so the second week, we will be learning about all of that, and we will be learning about, you know, big data in genomics, and genome-wide genetic variation. And then we will be learning about the example pipelines that are there. So I was telling you about pipelines, and I showed you genomics. So let's go back. Go back to our platform and look at the live example here. So here you will see that we have different omics data types here: transcriptomics, genomics, epigenomics, and then metagenomics. So in genomics, we will be looking at uh, mutation variants. We will be looking at these sort of pipelines, variant calling pipeline. So what is a pipeline? I asked you, what is a pipeline? So let me look at the chat and see if someone has, okay, that's great. I see some good answers. Perfect, yeah. Those are correct answers, all of you, almost. So thank you. Please, can someone in Agri Biosciences benefit from these trainings since emphasis on biomedical? Yeah, so the emphasis is not on only on biomedical, but we have a few examples from biomedical. data science, and we have a lot of examples from agriculture as well. So we have those projects, we have those projects that you can look into, and the technology remains the same. So learning about the technology and applying it to different bioinformatics research areas or research areas in general. So it can be agriculture or any of those. So as I was telling you that the pipeline, so we're going back to pipeline and workflows. So what are pipelines and workflows? Let's look at one. So we have created, like the Tauber Bioinformatics Research Center has created this amazing pipelines that helps us interpret complex data with ease. And also it let us learn about the technology. It tells us in these pop-ups that why and how to run this, what this means, and if there is any associated publication that we needed to run, that we needed to learn or read. So you kind of in a demo pipeline, what we have done is that we have used a publication based data set that we go through. So in a demonstration pipeline, we will be together learning this and doing this together. And you will be getting also video lectures to do and to learn to do how to know and learn how to do this. So we'll be sending you the courses here. You'll have access to the courses, which are hands-on courses on genomics, which describes these pipelines. And then we will be sending you the video tutorials. In addition to that, we will also have sessions. So this hybrid interactive program, Omics Logic programs, includes of all those components which I was telling you. So genomics would be the second week. So we will be doing genomics in the second week, and then we will be doing then we will be moving to metagenomics. So the second week will be about metagenomics. Okay, so do you guys can anyone tell me, or if any of you can tell us that do you have interest in metagenomics? Any of you? Have you heard of it, or have you thought of it ever? Like, what is metagenomics and where it can be applied and what is going on in this new field? It's a new field. So please let us know in the chat. Have you heard of this metagenomics analysis, microbiome, metagenome? It has a lot of application in, and we are also doing a specialized program that I'll show you on metagenomics. But this session and this week is dedicated for metagenomics. Okay, Valentina, thank you for telling us, and Toby also. So Valentina, you're interested in metagenomics. So this week we will be going through metagenomics and some of the projects that are there as a data set for learning metagenomics. So American Gut project is one of such project. So to those who are interested in learning about metagenomics, yes, this program includes metagenomics as well. So to learn about metagenomics, we will discuss about what microbiome is, how we can use next generation sequencing to study whole microbial communities and analyze individual microbial genomes. And we will also discuss data sets that are available for analysis of study of role of microbes play in what role microbes play in agriculture, what role microbes play in biomedical research, and epidemiological studies of infectious diseases. So microbes has a lot of application in today's world. And in this session, we will be learning about, microbiome composition and taxonomy. So in if I tell you about metagenomics is the field. It refers to both research technique and also a research field. So it can be defined as a genomic analysis of microbial DNA from environmental communities. So culture-free sequencing. So that's one of the biggest advantage of metagenomics. So in this, we will be learning about metagenomic data analysis, and you will have access to these three courses: Introduction to Metagenomics, and Metagenomics 2. So they cover from a wide range of metagenomics research. So anyone who has a focused interest on metagenomics, they can totally jump into the second week themselves from the very beginning. So if you're just interested in genomics, you can focus on genomics. If you're interested in metagenomics, you can focus on metagenomics, and you can reach out to us for any problems or any questions that you have, that you wanted to clarify about. So this program would be about that. And then after this metagenomics session, we have, which is on 17th February, as I was telling you, that we'll be doing 16S hypervariable regions, learning about those, studying NGS, gut microbiota, and how it relates to human health. Those are all covered in the metagenomics courses. And one of the biggest advantage of this would be that many of it would be depend on it would be on you to do. So you'll have your own time to do this at a time that is more convenient for you, because these will be emailed to you, and you will have access to the platforms for 1 month 24/7. So it depends when you take out your time, and you can learn from these resources, because they will be available for you all the time during this 1 month period when you're registered. So the last week would be on about transcriptomics, very important technology, and I think it's widely used for biomedical research, and also for clinical research and agricultural research, where we are identifying potential like highly expressed and low expressed genes in different forms. So we are sort of doing a lot of comparison. So it could be a mutant versus a wild type mutation, a comparison for agricultural stuff. So we have had a lot of projects where we used this sort of comparison for tomato, and also we have a project on crop drought, and and there is this one on potato. So we have a lot of projects that explains the cancer biology and biomarkers, how we can identify biomarkers by using transcriptomics. So this is, I think, a great opportunity to learn about different omics technology within 1 month, or maybe you can expand it to 2 to 3 months. It depends on you. So if you are wanted to focus on a specific technology, you can learn it for 1 month, and then extend your subscription for second month and learn about another technology. Or if you have time, then I would recommend you to go through all of them. That includes the courses. So in transcriptomics, we have transcriptomics 1, 2, and 3, which you will be learning from the introduction to um, RNA-seq, to applying RNA-seq, how data science and machine learning is being used to apply RNA-seq. So we have this session on 24th, and we will be meeting and discussing transcriptomics. And then, as I was telling you, that a lot of it is not only to learn but also to apply. So in transcriptomics 3, we will be learning about machine learning and how machine learning is used by applying them. So it will be, all of it would be hands on. So from data to different techniques, to different algorithms, to different programming languages. So that's the last part of today's presentation. So I'm going to take you to the platform again, and I am going to ask you all that if you have any questions, please let us know, or let me know, and we can actually answer this question and answer. So I see that a lot of people are sharing their email addresses. Okay, thank you, Tayo, for asking about the metagenomics. So we also have a dedicated program just for metagenomics that would be starting on 16th February, and we'll be sharing you the details. And this would be a really program for metagenomics research. So that would be really a cool thing for you to check out. So we'll be showing you that. But before that, I just wanted to show you that how this entire training will be conducted using these resources, these cloud resources that anyone can take. So transcriptomics, I told you that we will be covering transcriptomics and data science. So we have these 4 projects on transcriptomics that you can learn from already. And then we have this data science section, data mining section, where we have supervised and unsupervised analysis. So the supervised analysis, you will see that there are different algorithms that are applied to these sort of databases like random forest. These are machine learning algorithms, SVM, support vector machine, regression, and then discrimination analysis, a linear discrimination analysis, and there are different versions, and then testing. So there are different supervised analysis that we can perform on the data, and also unsupervised like PCA, clustering, if you have ever heard of these, and k-means. These are used for many other data sets as well, not only in biology but also in finance and all those other techniques, other data sets. So we will be learning from here, and then we have the courses. So I have been back and forth telling you that this entire process is quite simple, because you have tutorials to look at, and then you have a place to do your experiments. So this is how we are going to cover those true aspects. And once you get familiarized with all of this, and if you have an interest in Python and R, so can you tell me in the chat, do you have interest in Python and R, learning about programming or coding? Are you guys interested in coding, any of you? Okay, Fatima is interested in learning R. Ruby is also interested in learning R. Miriam is interested. Mansoor is interested. Prince is also interested. So, okay, thank you guys for letting us know. Clement is also interested. So next, I'm going to show you that uh, how the omics technology and the coding coding in bioinformatics, what we are doing, and how it is done. So we have created a platform just for you. I mean, people, for those biologists and bioinformaticians and students who does not have a background, to learn coding. So coding for bioinformatics. So this is not hardcore coding about R and Python. So I just want to tell you that there are a lot of free courses, and there are a lot of good courses also which are available to learn just coding. So this is not about learning just coding. It is about applying coding to bioinformatics, apply and do coding for these sort of data sets that we discussed. So these data sets, when we are working on them, and when we are running those statistics and algorithms, in the end, what we are getting some results that we want to visually see, right? We want to see them in a graph. We want to see them how they look, and how we can find a pattern from the data, right? So those sort of things are really one of the best features of these two programming and coding languages, which are R and Python. So what we have created is this Code Omics Logic platform. So let us all go to this platform, and Tayo, can you share it? Let me also try and share the link. And Sani, if you can share the Code Omics Logic link. So this platform is also free. So you can go there, go here and take free courses. So there are a lot of free courses that you can take from and learn from here. And if you're a part of the program, then you will also get some premium courses. Not sure about the process, but you can go to code.omicslogic.com. I'm just pasting the link here. Okay, the thing is pasted. Thank you so much. Yeah. So you can go to the coding platform and get started for free. So you can create a login right now here. So if you are there in this, if you have reached here, let me know. So if you are not logged in, it will look like this. It will be you will look it like this. Yeah, so there will be no courses here. And if you see the difference here, you will have courses here. So once you log in, you will see the courses, and let's check out the courses. So the courses are in a form which has code blocks. So all these courses will have code blocks where you can run your own code. So you don't need to install Python and R to your computer if you don't have a powerful computer, or if you don't have like a device. So you can do it from the browser. So this entire course can be done through a browser, and you can learn about all the NGS technologies. You can learn about coding. So let me show you how it is done. So getting started with bioinformatics, it's a free course. So the ones that have star are the premium courses. So it starts with loading data in R. So just the very first thing that you wanted to do. So once we start this program, we will be sending everyone the detail how to do this, like how to log into these different three different platforms to the participants who are joining. And we will be sending them more detailed tutorials to do this on their own also, so that they can look at it as a guide and do this by their own. So here you will be able to see that okay. So Adikunle, if you can mute yourself, please. Okay, thank you. Okay, thank you. So as I was telling you that it has different sections. The first section is to understand the problem. So we kind of explains what the this problem section is, what is this defined, then describes the code. So what is the code? You can read about the code details here, what it contains, and then you can download the data which uses this code, and then you can upload this code into this platform which will come next. So after learning, you know what is it and what is it is going to do, then you try it yourself. So here you have a code block that you can try yourself, and then you can run this code, and then you can generate different files. And I'm not going to do a demonstration right now because we're running out of time, but it's free and you can all try this up. So I will recommend you to go to this website and try this free coding lessons. And if you join, then let us know. We'll be able to help you more with this. So this is the coding. Okay, so one of the things in the end that I wanted to add is that how would we know that what are you doing? Because it's an online program. We are not sitting with you. We are not knowing that how much you have learned. So what do we do? How do we know that how much you learn? So we know by looking at the activity. So all the activity that you do, all the learning that you do on the three platforms, you can see your performance. You can see how much you have finished the courses. Everything is there in your profile. So it let you know that how much you have learned. It let us know how much as a group you guys have learned. So we can see that you can see, for example, here there are sessions and there are projects. So this is the activity on three platforms of all the users during this time, what they have done and how much they have learned. So for this course completion, we expect every one of you to get at least 3,000 points in 1 month. So the point system does work with the number of courses like you finish, all the activity that you do, and also like get a profile. So for here, you can see an example, like once you finish the courses, once you complete some of the tasks which are like running the finishing the assignment and stuff, you will get points, and then you will be able to learn from those to next to the projects, that how it is applied to the projects. And we have had like this previous program that we did in September in October. So we have had a lot of reviews and feedbacks that we got from participants, especially from Lagos and especially from Lead City and other nearby cities there. And we got a lot of students who really went through the entire exercise and actually achieved a lot. So we have had several students who came from different backgrounds. So not just biology or microbiology, but different backgrounds. And they were able to do finish these courses and learn from it. So the background and the other problems that they had were were solved, and they were really happy about this training, that what they have learned. So as I was telling you that how do you apply this into a real research? So we have an ongoing program. Today we had the first session, the second session, sorry, about this program. So which is the infectious disease program, where we are learning and applying this technology, especially for infectious diseases. So we are just working on corona research, COVID-19 research, Ebola research, tuberculosis and malaria research. And the group especially that we are working in Ghana is Noguchi Memorial Institute for Medical Research, NMIMR. And there we have a group of scientists who have interest in their in bioinformatics, and they wanted to do this for vaccine research. They are doing it for drug repurposing, and they're looking at population level data. So we are working together in collaboration with them, and many of the students who were interested in infectious diseases, PhD students or faculty, those who wanted to do this sort of research and have some sort of either grants or they do have some financial support, they joined our program for this. So this program has started, and if any of you are interested in learning more about this program, please reach out to us. Please reach out to africa@pine.bio. So Tayo, if you can just put that your email address, and you can also reach out for us for the Metagenomics program. And I saw that many of you have shared the Metagenomics program details that you are interested in. So this one is going to start from 16th February, and all of our programs, these research programs, have an option of scholarships. So you can obviously apply for scholarships as well, because these are programs that would require you to have a research license and upload data and do those analysis. So I welcome you all to join any of these programs, but I think it's great if you're a beginner to start with this this program that we discussed today on basic bioinformatics, basic omics logic, in collaboration with MedlabConvo and our team at in Nigeria. So I would like to now pass it on to Tayo to discuss a little bit more about what his practical experience has been and how we have been supporting together this initiative.
Hello, thank you very much, Mohit. Thank you for the wonderful expose. Well, presently, there is also another introductory class on basic omics logic. And from my own experience from this end, the Biobiotech Omics Logic program is a well-structured, well-organized training logic programs for beginners, for people who are novel, who have no prior information or prior idea about bioinformatics, whether as students, researchers, or as clinicians. I'm from a clinical background. I'm actually a dentist. And with Biobiotech platforms and educational resources,
Thank you, Tayo. And
I have been able to get grounded, that would not have won before joining. Clearly, the practical hands-on program. Of course, we do have the asynchronous courses on the educational platform on the edutibao. We have suggested, we have employed you to join. We do have the introductory courses there, but aside that, the pipelines, the coding platform, and the virtual lessons are majorly practical hands-on programs. This is not a program where you only come here to listen to the theory and listen to courses. At each program, there will be practical hands-on that will expose you to either coding or to either the server-t-bio.info pipeline, where you can run analysis, you can visualize your sequence, visualize your read, and visualize mutation pipelines. Likewise, that you can also do the same thing with metagenomics and microbiome data. So I will advise you to log in and avail yourself of this opportunity. And I know this, even though this is one under the auspices of MedlabConvo with Aisha, Diola, and Emmanuel, I'm sure you have the best hands and the best mentors, both locally through the MedlabConvo team, also through the OmicsLogic Nigerian team, and internationally through our international team led by Ilya Brutsky, who is the CEO. And we also have Dr. Aprit Kaur, who has a PhD. We also have Dr. Mazonda, who has been discussing with us since the beginning of this webinar. So this is an opportunity to really log into, to explore cutting edge research technologies, to learn data analysis, to learn bioinformatics, and to be able to help you forge ahead in your research, in your postgraduate studies, in your undergraduate and postgraduate studies, and in your clinical discipline. I will enjoin every one of you to please log in and avail yourself of this immense opportunity. And I'm sure you are not going to regret it. This is something that can catapult your career or your graduate studies. You definitely have testimonies afterwards. Please join us as we build an amazing community of life science citizen scientists who think globally. The coronavirus pandemic has opened our eyes and ears to the immense opportunities in genomic studies, in the multi-omics integration studies. And this program will not only introduce you to them, it will also guide you step by step on how to become grounded in bioinformatics and life sciences. So please join us, and you will have testimonies at the end of the program. And it's not a one-time program. I'm sure MedlabConvo and other partners in the OmicsLogic program are planning a series of programs, so that it's not only that we make you a beginner, we also take you through, because I'm sure we also have intermediate courses, advanced courses, so that not only will you learn, it's a continuum. It's not a one-time thing. It's a continuum where you learn, where you're grounded, and by the end of this series, you will be able by yourself, be able to run these pipelines and do these research projects to publication stages. And at any point in time, you have mentors to guide you through all the stages. So once again, thank you for joining us today. And I'm sure you'll be part of this novel, unprecedented, or well-established community. Thank you, everyone.
Thank you, everyone. And thank you for joining today's session. And we also have Aisha. So Aisha, do you want to say a few words and conclude the session?
Okay. I am Aisha Diola from MedlabConvo in Nigeria. And it's been a wonderful session. And I hope everyone that participated and joined have really gained a lot, and we look forward to having everyone enroll for the courses and apply them into various aspects of their field. Thank you very much, everyone. We really appreciate you all.
Thank you, everyone. And we will see you soon. Have a nice day. Take care. Bye.